Okay, it's been about an hour since the yeast have been trying to use either the Splenda or the sugar. I would like you to make a hypothesis before I show you the results. Do you think the Splenda was used as well as the sugar by the yeast? And we'll be able to tell that by the size of the balloons. If they used each as well, the balloons will be the same as in size. If the yeast did not like the Splenda, the balloon will be smaller than the balloon for the sugar. So what do you think happened? Make a prediction before I show you the results. Okay, here are the results. As you can see, the Splenda is almost the same size as the sugar. That might be surprising. It's not quite as big as the sugar, but it's about the same. I could take a tape measure and measure that for you, but you probably can see that the sugar is a little bit bigger. You can see that the control has no gas in it, so the yeast had nothing to eat, so they could not undergo cell respiration. The Splenda had some gas produced, so the yeast can use that sweetener for food. So sometimes when you make baked goods and you want to use a sugar-free baked product, you can give the yeast Splenda. It just won't produce as much gas as maybe the sugar does. And the sugar, you can see, or the sucrose, which is actually made of glucose and fructose, has a little bit more gas in it than the Splenda does. Okay, write up your report now. In your conclusion, make sure you understand if your hypothesis was supported, any errors that occurred that could have caused inaccurate results, and then anything in the future that you could try. How could we change this experiment so that we could find out more about cell respiration?